Bom dia a todos. Good morning. I would like to thank Professor Bergonzi for having accepted our invitation for a lecture here in our course of pharmaceutical science. And I will introduce Professor Maria Camilla. Is everybody hearing me? Vocês estão me ouvindo, pessoal? Okay. Professor Maria Camilla Bergonzi is Associate Professor at the University of Florence, Italy. She has a deep knowledge in the improvement of bioavailability and technological features of bioactive molecules from plant matrices and drugs by using conventional and innovative drug delivery systems. Her studies are concerning the preparation of delivery systems able to cross the blood-brain barrier and promote the oral absorption. She is titular of course of Pharmaceutical Technology and Legislation for ungraduated students of degree course in pharmacy and co-titular of the course Soft Matter Materials Applied to Drug Delivery Systems, Food Supplements and Cosmetic Science for the master degree in Advanced Molecular Science. I don't know if it's all correct, Professor?
Yes, thank you very much for the presentation, Professor Conrad.
Okay.
Thank you.
Then let's hear from you, your lecture, please. Be welcome.
Okay, thank you very much. Good afternoon, everyone. I share my... Can you share my screen?
It's okay.
Okay. It's okay. Thank you very much, Professor Conrad, for giving me the opportunity to present, to show you some results performed, obtained in my laboratory in Florence. My laboratory is a PhytoLab, is a laboratory that works with natural compounds for vegetable origins. There are growing interest in single molecules on extract of plant origin by patients and healthcare professionals in the last decades, because natural compounds are compounds with many chemical structure, with many topics, many targets inside the cells, into the tissue, the organs, and these molecules can represent new strategies to prevent or treat diseases. Is also the natural compounds are useful tools to reduce the side effect related to conventional therapies. The population uses natural products as a drug, but also as cosmetics, as food, as medical device. But natural compounds, single molecules, but also extract, show some drawbacks, some limitations. In particular, we have a very low solubility, aqueous solubility. It is aspect to reduce viability of these compounds. Natural compounds present high metabolism, liver metabolism, and also low permeability across the biological barrier, in particular blood-brain barrier, skin barrier, but also intestinal barrier. Nanomedicines represent a useful tool to increase the viability, biopharmaceutical aspect of natural products, because a nanovector provide a high surface area and improve the solubility of the compounds, overcome multi-drug resistance of natural compounds, and increase drug efficacy. In my laboratory, PhytoLab, we applied the nanocarrier, nano-delivery system to deliver of vegetable compounds or extract for improve some aspect, biopharmaceutical characteristics, in particular increase solubility, but also increase permeability, in particular in vitro and also in vivo permeability across the barrier, improve the solubility, but also the stability of the molecules in different media. With nanocarrier, it's possible also obtain a modified release of the drug inside the body and reduce the toxicity. Many are the drug delivery system applied to natural compounds. We can use a vesicle such as liposome or niosomes, nanoparticles such as magnetic, gold nanoparticle, polymer nanoparticles, but also micelles or lipid carrier, similar solid lipid nanoparticle or nanostructure the lipid carrier. It's possible also obtain a modification of a surface of nanocarrier to obtain a multifunctional carriers. Multifunctional carriers as nanocarrier where the surface are modified with a different structure. In particular, we can obtain in this way a targeting of action of the drug by inserting on the surface particular ligand similar antibody or polyethylene glycol, carbohydrate, peptide, folic acid or transferrin to targeting the action, the drug loaded inside the nanocarrier, in particular cells, in particular tissue, in particular organs. For example, if on the surface we introduce hydrophilic surfactants such as polysorbates, polyethylene glycol, poloxamer, and so on, it's possible to obtain stealth on long circulating nanocarrier to prolong the bloodstream, the time, the circulation time of the carrier and then of the drug. The selection of the carrier is related to the characteristic of the drug, or the extract in the case of natural compounds, the release that I want to realize, and in particular prolonged release, controlled release, the therapeutic target, the route of administration, the possibility of sterilize the carrier, but also the scale up of the production of the carrier. Now some characteristics, some nanocarriers used in the delivery of natural compounds. First of all, nanovesicles. In particular, we start with liposomes. Liposomes, you know, are the phospholipid bilayer. The bilayer is similar to cellular membrane, is made of phospholipid and usually cholesterol or other lipids, and the bilayers delimit aqueous phases, one or more aqueous phases, aqueous compartment. An outer vesicle are ethosome and transferosome. These are carrier important for the skin delivery because ethosome with the presence of ethanol or transferosome. Transferosome is a vesicle where is present at edge activator, for example, polysorbate, and produce a vesicle, ultra-deformable vesicle to improve the penetration across the skin. Niosomes are liposomes produced by synthetic tensides. Liposomes are carrier biocompatible and also biodegradable, related to the constituents of the bilayer, can load both hydrophilic and hydrophobic molecules, but also extract, reduce the toxicity of encapsulated drug, are vesicle with high flexibility and versatility of use. In this graphic, there is a possibility different mechanism of penetration of liposomes inside the cells. There is a mechanism by fusion, by endocytosis, but also diffusion of the drug from the vesicle inside the cells. About the lipid nanocarrier, we have solid lipid nanocarrier, nanoparticles, SLN, and nanostructured lipid carrier, NLC. Solid lipid nanoparticles are solid nanoparticles at room temperature, and the core of the carrier is made of solid lipid. Around there is a layer of tenside. In the case of NLC, inside the nanoparticles are present solid and liquid lipid. The presence of liquid lipid improve the load capacity of nanocarrier and also improve the stability of the carrier, because in the case of SLN it's possible have expulsion of the drug during the storage. Also in this case, these are solid carrier, and there is in this case the possibility to obtain a powder with the freeze drying process, and these carriers can be introduced, for example, in the hard gelatin capsules or in the food to guarantee a prolonged release of the drug, to stabilize also the drug, because are carried with a longer term stability, and related to the constituents of the core, the solid or lipid liquid selected, we obtain biocompatible and biodegradable materials. Another nanocarriers are the nanoparticles, in particular polymeric nanoparticles made of natural or synthetic compounds. We have inside the nanoparticle nanosphere nanocapsule. Nanosphere have a matrix type structure, and inside the nanosphere we have the drug as a solid or liquid. In the case of nanocapsule, we have a core, a reservoir of the drug, and around we have the membrane that control the release of the drug. Here I reported some polymer used for the preparation of nanoparticle for human application. For example, polyethylene glycol, chitosan, gelatin, casein are polymer, natural polymer used to produce these nanoparticles, but also copolymer, biodegradable and safe compounds similar to polylactic glycolic acid, PPGA, polylactic acid. Also, there is also synthetic polymer come polyepsicaprolactone or butyl chanacrylate derivatives. Other nanoformulations important for delivering particular extract, because these nanoformulations improve the solubility of different kind of molecules, are microemulsion. Microemulsions, oil in water, water in oil, is a clear, transparent system, optically isotropic and stable, where there is a lipophilic or hydrophilic phase surrounded with a high amount of the surfactant. In this case, this formulation improves the solubility of the plant-based extract due to the presence of oil, but also of the high percentage of tenside around the inner phase. Another carrier are polymeric micelles. Polymeric micelles are self-assembled core shell nanostructure. These micelles are made of amphiphilic block copolymer. In the amphiphilic polymer, we have an hydrophilic portion and hydrophobic portion. The hydrophobic portion above the critical micelle concentration produce a core, hydrophobic core. Inside this core is possible also the lipophilic drug, and the core, around the core, there is a hydrophilic shell. The dimension of these micelles is between 10 and 50 nanometer. It's possible to realize due to the different polymer selected to produce these micelles, or passive targeting, and then with APR effect, we can administer the drug in the inflammation tissue or cancer tissue, where there is the possibility to enhance the permeability and retention effect, or derivatize also in this case the surface of micelles with sugar, with peptide, with antibody, to obtain a targeting or action. Some word about the characterization of nanocarrier. When we prepare a nanocarrier, a nanoformulation, first of all, we performed a physical characterization by dynamic light scattering or electrophoretic light scattering. With DLS, we define the mean diameter of the carrier. Then the nanocarrier is about 10, 20, 50 nanometer. The dimension are important for the route of administration. If we prepare a nanocarrier for parenteral administration, the dimension is around 200 nanometer. Also by DLS, it's possible to characterize the homogeneity of the samples by polydispersity index or polydispersion. By electrophoretic light scattering, we define the potential, the zeta potential, and the charge present around the particles. The presence of high charge, positive or negative, the high potential around the particle is important to stabilize the system, to guarantee the stability of the colloidal dispersion. The morphological analysis is performed with the transmission electron microscope and confirm the DLS data, the shape, the aggregation of the nanocarriers, the dimension of nanocarriers. Then there is the chemical characterization. In this case, we used a chromatographic method with detector, fluorimeter, or DAD, and we define with the chemical characterization the amount of the drug inside the nanocarrier. We can define encapsulation efficiency, which represent the drug inside the nanocarrier with respect to total drug, or the load capacity, the drug inside the nanoparticle with respect to weight of the nanoparticles. Also, we characterize the nanocarriers by monitoring the release of the extract of the molecules from the nanocarriers to study if the release is prolonged release, sustained release, and so on. And we compare the release of the drug from a solution from nanocarrier. Also, we performed the stability study in different storage conditions, for example, different temperature, or we use the freeze-drying process to improve the stability of the carrier. In my laboratory, also, we studied the influence of the nanocarrier on the in vitro permeability of the drug. Then we compare the permeability, the in vitro permeability of the drug, and the in vitro permeability of the drug formulated in nanocarrier. We can use a Pampa test, permeability. This is a test that monitors the influence of carrier on the passive permeability. We have a donor compartment, an acceptor compartment, and an artificial membrane. This artificial membrane was derivatized to produce membrane barrier similar to physiological, biological barrier. For example, to mimic a gastrointestinal membrane, we derivatize the artificial membrane with lecithin and cholesterol. In the case of skin, we used silicone and isopropyl myristate. In the case of BBB, blood-brain barrier, the artificial membrane is derivatized with a porcine polar brain lipid. Also, we studied the influence of a nanocarrier not only on passive mechanism, but using CACO2 cells or BBB cells, we study the influence of the nanoformulation on the active mechanism involving in the permeation of the molecules. Now, some examples, some formulation used to improve the biopharmaceutical characteristic of natural extract or vegetable extract or vegetable compounds. One study is related to do the formulations of 3D extract, in particular Vitex agnus castus, used in premenstrual syndrome, Salicis cortes, known for analgesic, anti-inflammatory, antipyretic activities, and also Silybum marianum. The phytocomplex of Silybum marianum is silimarin, and silimarin is used for disease of liver, spleen, and silimarin is active against cancers, and also has been found to be beneficial in type 2 diabetes patients. Here are some constituents of phytocomplex of poultry extract. In the case of Agnus castus, we have iridoids, agnuside, and flavonoids. In the case of Salicis cortes, we have salicin, salicortin, and flavonoids. Silimarin is characterized by flavonoid, in particular taxifolin, and flavolignans, silibinin, isosilibin, silicristin, and silianin. The characteristics of these extracts are poor oral solubility, oral viability, poor aqueous solubility, and high instability. For the extract, we select as formulation macroemulsion. Macroemulsions are a transparent system with respect to emulsions, and to select the best compounds to produce macroemulsion, first of all, we studied the solubility of phytocomplex in different oil and different liquid tenside. We selected for each extract different component that improve the solubility of phytocomplex. By using the pseudo-ternary diagram, we obtain the percentage of different constituents to have a region of macroemulsions. In particular, this is the receipt of macroemulsion for Angioscastus, triacetin 20%, Cremoforel tenside 22%, Labrazole 22%, and water. The constituents are both tenside, are also solubilized agent, and some of the compounds selected in the formulation are also penetration enhancers, then can improve the penetration, the absorption across intestinal barrier. Considering the solubility, with macroemulsions, with optimized for macroemulsion, we have a good results in improved the solubility of extract. We have an improved solubility of 10 times in the case of Angioscastus, 3 for Salicis costes, and 40 times increased the solubility for Silimarin. About the influence of a formulation on the in vitro permeability, we performed the Pampa test and also the test in the presence of CACO2 cells. CACO2 cells produce a layer similar to intestinal barrier, then there is the cell similar intestinal cells in the layer, and we calculated the permeability coefficient, PE in the case of Pampa, and P-up in the case of CACO2 cell study, and we observed that the formulations improved the in vitro permeability for all 3 extracts with respect to extract in water. Then a good solubility not only about related to the solubility, but also the in vitro permeability, and this result is related to the selection of the component of macroemulsions. Lipid tenside are not only solubilizer, but also PGP inhibitors or solubilizer and penetration enhancers. Here, a study related to the delivery of Silimarin by using nanostructural lipid carriers. In this case, we prepare 2 different NLC. One NLC contains Silimarin and stearic acid as solid lipid, Capriol 90 as liquid lipid, and Bridge 78 as an anionic surfactant. This surfactant is characterized of the presence of a pig moiety that prolongs the circulation time of the carrier. Another, the second NLC I formulated to improve the characteristic of Silimarin extract, contains acetyl palmitate as a solid lipid, lauroglycol 90 for liquid lipid, and Bridge 78 as an anionic surfactant. We characterize from a physical and chemical point of view the carrier. In particular, here is the carrier, the NLC with stearic acid loaded with Silimarin. We obtain a dimension around 200 nanometer. This formulation is for oral administration, as the dimension is good for oral administration. This system results homogeneous, PDI less than 0.3 produce indicate a homogeneous system, and also the zeta potentials, the potential around the nanoparticles, around the NLC is high, and this stabilizes the system, the colloidal dispersion. Both formulations shows a very high encapsulation efficacy of the extract. With these 2 formulations, we have an improved solubility, aqueous solubility of Silimarin, but also a very high improved permeability across the artificial or cells membrane. To discriminate the best formulation for deliver silimarin extract, we performed in this case an in vivo test. In this case, the formulation were tested in a mouse model of induced metabolic syndrome. The metabolic syndrome was induced by administering high-fat diet, and also we induced a type 2 diabetes in the mouse, and then a hyperglycemia by using streptozotocin. From the literature, silimarin has good results in reduce the blood glucose levels and also the triglyceride levels. Then we administered in the mouse silimarin alone and also our two formulations. In one group, NLC with stearic acid, another group, silimarin loaded in NLC with cetyl palmitate. We monitored during 1 month the blood glucose level, triglyceride levels. Here are some results. In particular, both formulations are active to reduce glucose and triglyceride levels, but the NLC with stearic acid, the treatment with this NLC reduced the glucose levels and the triglyceride levels during all the time of the experiment. Then maintain low levels of glucose at the level of triglyceride for 1 month. And then in vivo study showed the best performance of this formulation with respect to NLC made with cetyl palmitate. Another study is related to European project in which I was involved. In this European project, I have to formulate, to prepare nano formulations to improve the pharmaceutical characteristic of different extracts obtained from olive leaves. The project aimed to valorize waste from olive leaf production to obtain products for pharmaceutical, cosmetic, or food interests. In particular, I formulate an enriched extract containing oleanoic acid by 98% and an extract containing triterpenes. The problem of both these matrices is the low solubility. In particular, the solubility of oleanoic acid in water is less than 1 microgram per milliliter, and also we have a very low solubility of the triterpene extract, about 7 microgram per milliliter. Also, in this case, the microemulsion results very good formulation because we developed 6 different microemulsions for the delivery both of oleanoic acid or triterpenes. We obtain an improved solubility until 1, 2, or 3 milligram per milliliter or of oleanoic acid or extract. Also, in this case, by Pampa test, we checked that the microemulsions improved the permeability, the passive permeability of oleanoic acid of triterpenes. Here, the receipt of different macroemulsions developed. Macroemulsions are liquid, transparent formulation, and it can be commercialized as liquid, or it's possible also insert the macroemulsions inside our soft gelatin capsules. We use it when we prepare nanocarrier, macroemulsion, nanoparticles, and so on. We selected excipient component defined the GRAS safe from FDA, and also in this case we used compounds excipient used for human administration, in particular for oral administration. Some of these compounds are synthetic compounds, synthetic excipient, and are solubilizer, are penetration enhancer, and some of these compounds, for example, TPGS, a derivative of the alpha tocopherol, is a compound that inhibits PGP and then can improve the permeability, the absorption of the loaded drug. We performed many in vitro in vivo study with these formulations. In particular, I will show you the results of this study. In this study, we selected these formulations. In particular, this formulation, this macroemulsion, is able to load 2 milligrams to improve the solubility of triterpenes until 2 milligrams per milliliter. And we tested the extract and also the macroemulsions on lipid accumulations induced by high glucose condition in HC2 cells. High glucose levels, as reported in the literature, improve the development of tumor and increase intracellular lipids. Also in the literature is reported that oleanoic acid, the main constituent of our extract, inhibits the progression of hepatocellular carcinoma in HC2 cells. Then we want to test the efficacy of the effect of the extract that contain oleanoic acid but contain also other terpenes, and also the effect of the extract loaded in macroemulsion. Then we monitored the effect of these two samples on the lipid accumulation in the hepatocellular model. We treat the cells with high amount of glucose, as reported in literature, and we observed an increase and improved the production of lipid inside the cells. The lipid accumulation was evidenced by a colorimetric method, Oro method. And when we apply different concentration of a triterpene extract on the cells with high amount of lipids, we observe a reduction of lipid accumulation. In particular, this reduction is dose-dependent effect, and we observed that in the case of extract 0.3 microgram per milliliter, we have a reduction about 35% of lipid accumulation. If the extract is formulated in macroemulsion, as shown in this figure, we have a reduction about the 50% of the lipid accumulation. Then, with the same amount of the extract, but in presence of the microemulsions, the efficacy of the extract is improved. This is due to the presence of the constituents of the macroemulsion that improve the penetration of the extract inside the cells. Another compounds that we formulate in different nanocarrier is thymoquinone. Thymoquinone is a monoterpene, is the main constituent of essential oil or fixed oil of a plant, Nigella sativa or black cumin. Many pharmacological activity are reported in the literature about the thymoquinone, but the molecule has some limits, in particular low solubility, instability in different pH, photosensitivity, and thermosensitivity. In literature, there are many nanocarrier developed for the delivery of thymoquinone. We developed a mixed micelle formulations. In this case, we produce a polymeric micelles using soluplus and solutol in the ratio 1:4 to produce the micelles. Both the polymer are amphiphilic polymer, and the hydrophilic portion is represented by polyethylene glycol. These compounds, these polymer are solubilizer agent, but also solutol is a PGP inhibitor. The micelles have a good dimension, about 50 nanometer, and high encapsulation efficiency with respect to thymoquinone. We test these micelles loaded with thymoquinone in neuroblastoma cells to evidence the influence of formulations on the inhibition effect of thymoquinone on cell migration. From the literature, thymoquinone is anti-proliferative compounds, inhibits the development of a tumor. In this case, we tested with the scratch test the influence of thymoquinone, and in particular the formulations on the cell migration of neuroblastoma cells. We can observe in the figure A that here we have a layer of cells, and we produce a scratch inside the cells. We observed the reduction of scratch during the time. Here is the situation with the control. We tested also the mixed micelle without thymokinone to verify also the influence of the empty carrier on the action of the migration, but no effect was evidenced. Thymokinone reduced the migration because at two different concentration, but the influence, the efficacy of thymokinone is enhanced when the thymokinone is present inside the mixed micelle, because the scratch, the closure is bigger in this case in the present formulation with respect to drug alone. Then also in this case, the carrier, the nanoformulations improve the bioactivity of thymokinone in this in vitro test. These cells with these formulations, we performed also in vitro study about the influence on in vitro permeability. And in this case, we monitored the influence of mixed micelle on permeability across CACO2 cells to mimic intestinal barrier, but also this type of cells that are cells of human blood-brain barrier. And in both cases, the micelles improve the permeability of thymokinone. Also, we performed a study about the toxicity and the biodistribution of these micelles, thymokinone-loaded micelles, inside zebrafish embryos. Zebrafish is a transparent fish used usually for the study of toxicity, biodistribution of the drug inside the fish. The fish has a similar barrier, similar tissue organs than human, and in this case, we use it to do different fish, wild type, a transgenic line. In this transgenic line present a cardiomyocyte marked with green fluorescence protein, and we evidenced that at low concentration, the micelles are not toxic. The micelles are a little bit toxic at high concentration. We have observed, for example, a problem with embryos. We have cardiac ventral edemas, a modification of axial tail, and in the case of transgenic line, we observe an improved modification of frequency of the heart. No modification are present about the capillary. Another formulations performed, developed for the thymokinone deliveries was liposome. In particular, in this case, we used for the preparation of liposomes phosphatidylcholine and cholesterol, but also we developed also the liposome where the bilayer was coated with hyaluronic acid. Hyaluronic acid is a polymer, a biocompatible polymer with gelling properties, adhesion characteristic, but also anti-inflammatory properties. We developed 4 formulations with different amounts of thymokinone, 2 liposomes without the coating and 2 liposomes with coating. We the same way we characterize the carrier for physical and chemical point of view. We study the release of thymokinone from the carriers, and we observed that the liposome realize a prolonged release with respect to solution, the release of thymokinone from a solution, and in particular the presence of hyaluronic acid around the carrier, in this case, produce a reduction of release of the drug. This is due to the presence of hyaluronic acid, and because there is first an hydration of the coating and then a release of the drug. This hydration process produces a low release of the thymokinone. The formulation result stable. In this case, we show you the physical properties of the system. Storage 4 degrees centigrade for 5 weeks, and we observed that the size and PDI zeta potential remain constant. We tested these formulations in preclinical models of tendinopathy. This because hyaluronic acid is excipient, a polymer with anti-inflammatory anti-inflammatory activity, and also used in osteoarthritis or tendinopathy to restore the tendons, to reduce the inflammation, and also to improve the production of collagen. Then we prepare this liposome also to improve the anti-inflammatory activity of thymokinone. After the administration, we produce the inflammation by injection of carrageenan. Then we tested the efficacy of liposomal formulation following 5 peritendon treatment performed at the days 1, 3, 5, 7, and 10. The tests are behavioral tests, and at a different time, my colleague pharmacologist monitoring performed paw pressure test to evidence the pain threshold of animals in response to noxious and non-noxious stimuli. Also to check the motor coordinator rotarod test was performed, and also the incapacitant test. In gray, there are the results related to the formulations with hyaluronic acid. We tested also one of the formulations without hyaluronic acid, the carrier without the drug, and also the drug, the thymokinone alone without the formulations. The results, the presence of hyaluronic acid coated enhanced the anti-inflammatory effect of thymokinone with respect to thymokinone alone. Then also in this case, the formulations ameliorate the bioefficacy of the molecules. The liposome containing high doses of thymokinone, in particular 2 mg per milliliter, and covered with hyaluronic acid results the best formulations. The anti-inflammatory effect is due to the presence of thymokinone, but also the anti-inflammatory effect is due to the presence around the liposome of the presence of hyaluronic acid. The hyaluronic acid restore the tendon damage and improve the matrix organization, the tissue organization. This is also evidenced by histological study. We prepare also another type of liposome for the delivery of thymokinone. In particular, in this case, the bilayer is made of phosphatidylcholine and fluorooleic and liquid lipid were used instead of cholesterol. Also, in this case, we cover the liposome with hyaluronic acid. We test, in this case, the formulation in conjunctival and corneal cells. As evidenced by this figure, the presence of liposomes, but in particular the presence of hyaluronic acid, improve the uptake, the cellular uptake. This is due to the presence of the CD44 receptor on the surface of the cells. These receptors are tagged of the hyaluronic acid. Hyaluronic acid is used also for target this system, this receptor on the cells for improve the uptake, the cellular uptake. Also, we observed that the presence of liposome around the thymokinone reduce the inflammation characteristic and of the toxicity of thymokinone reported in literature. From team analysis is possible evidence that the presence of a layer around the liposome. These liposomes are also tested in a model of dry eye pathologies. And in this case, we observed that thymokinone, as reported in the literature, reduce the inflammations, reduce the inflammation in the dry eye model, but also the presence of liposome and of hyaluronic acid around of liposome ameliorate the efficacy of thymokinone against the inflammatory produced in the dry eye. Another study is about another natural compounds, such as usnic acid. Usnic acid is one of the main constituents of lichens, in particular Usnea lichens. The molecules, as reported in the literature, has many pharmacological activity, but has some biopharmaceutical drawbacks, in particular low solubility, low stability, low permeability. For the delivery of using acid, we prepare polymeric micelles by using solvent evaporation method, and these micelles are made with solubles, solutol, and in this case we add TPGS. TPGS is a soluble derivative of tocopherol with solubilizer and PGP inhibitor characteristics. In the presence of the formulations, micelles improved the UA solubility about 300 times, because we obtain a concentration with the micelles of about 1 milligram per milliliter of dispersion. And also, we decided to prepare a gel. Then for prepare topical formulations. Then we prepare, first of all, the dispersion, colloidal dispersion of micelle that contain using acid, and then we gelify this formulation using a Sepigel 305 as a gelling agent. This is a synthetic gelling agent that produces at 1 or 2% a very good stable gel, and the constituents of the Sepigel is a polyacrylamide, a isoparaffin, and LRT7. Then Sepigel contain a lipophilic phase, but also a tenside inside the formulations. We prepare different gel with 1 or 2% of Sepigel in water, and we add the micelles. We characterize the micelles and also the gel using light scattering technique. We obtain a very small size of micelles inside the gel, a good systems with very homogeneity and high zeta potential, but also we characterize for the viscosity. Then by rheological point of view, the gel, we obtain that related to the viscosity of the gel, the system is a pseudoplastic systems. Also, the gel was tested in a texture analyzer to characterize the spreadability of the gel, the adhesion of the gel important for adhesion of the skin, and we obtained the results that showed a good spreadability and a good adhesion of the gel, but also a easy remoction from the skin. During the physical stability test, we characterize the gel for a rheological point of view. We check at different time the rheological behavior of the gel and also the water loss during the storage. We store the gel containing micelles with using acid at 4 or 25 degrees centigrade, and at different weeks we weight the gel to evidence the the water loss. We observed from the results that the gel is stable, and we have also a little water loss at 25 degrees centigrade after 1 month. We study also the release from the gel of the usinic acid. We compare the release of usinic acid from the system micelle in gel and also usinic acid in gel. For study this release, we used the France cells. We observed that in the case of micelles incorporated in the gel, we have a high release of the usinic acid, and different with respect to how dispersed the usinic acid dispersed in gel. And also with micelles dispersed in gel, we have a faster release and high amount of the drug released after 6 hours with respect 1 day in the case the usinic acid dispersed a solid in the gel. Then we improved the release with the formulations, macroemulsions and gels, and also improved the quantity of the drug released during the time. We tested the micelles loaded with usinic acid, but also the gel that contain micelles with usinic usinic acid in the in vitro for the activity on Staphylococcus epidermidis. And the results showed that both micelles and gel maintain the antimicrobial, antibacterial activity of usinic acid. We consider for activity a reduction of vitality about 5 logarithm of a reduction of bacteria cells of 4 logarithm. And in this case, we observed that both polymeric micelles and also the usinic acid in the topical formulation has the same antibacterial activity than usinic acid alone. The last one study is silibinin loaded in a nanoparticle made of ellenoic acid. In this study, we used ellenoic acid to produce nanocarrier. Inside the nanocarrier made of ellenoic acid with loaded silibinin. Silibinin is the phytocomplex of Silybum marianum with many pharmacological activity, anti-inflammatory, protective, chemopreventive, and so on, but has a very low solubility, aqueous solubility. Ellenoic acid, used as a carrier in this case, can self-assemble to produce nanoparticles. Ellenoic acid is the main constituent of olive leaves. Also, these compounds has many pharmacological activity. And then we use ellenoic acid for its ability to produce nanoparticles, but also for its pharmacological activity. In the literature, there are many studies that reported that natural triterpenoids, such as ellenoic acid, ursolic acid, or glycyrrhetanic acid, can self-assemble in nanoarchitecture, in nanoparticles, due to the presence inside the molecules of hydroxyl, carboxyl, or chiral center. Here, in this figure, there is the ability of ellenoic acid to interact and to produce after self-assembling nanoparticles. Nanoparticles are due to the presence of hydrophobic interaction between the molecules of ellenoic acid, but also hydrogen bond interaction. We produce nanoparticles using emulsion evaporation of organic solvent, and for stabilize nanoparticles, we used polyvinyl alcohol. The silibinin inside ellenoic acid nanoparticles improved the solubility until 0.27 milligram per milliliter, and the physical chemical parameter of this nanoparticle are very good parameter because we have size around 200 nanometer, a good homogeneity of the samples, and zeta potential, and a good zeta potential and a high encapsulation efficiency of silibin inside the nanoparticles. For this system, we tested also the possibility to obtain a dry product by freeze drying process, and we obtain a solid formulations, and after the addition of water we obtain again the colloidal dispersion of the silibin nanoparticles. With the Pampa test, we observed that the nanoparticles of ellenoic acid improved not only solubility of silibinin, but also the permeation, because with nanoparticles we have a very high amount of silibin permeate with respect of silibin alone, and a good results about in vitro permeation. In this case, we performed also a molecular modeling study to evidence the interaction between silibin inside the nanoparticles with ellenoic acid. The interaction are due to the hydrogen bonds, pi-Y interaction, but also van der Waals interaction. The molecular modeling prove the stability of the system, and also show a low release of silibinin from the nanoparticles, as also evidenced by our in vitro release study. These nanoparticles are studied for their anti-cancer activity, and the cytotoxic activity of silibinin loaded in oleanoic acid nanoparticle was considered on CACO2 cells. In this case, CACO2 cells are considered our cancer cells. And from preliminary results, we obtained that silibinin loaded nanoparticle reduces cell viability and then show cytotoxicity activity with respect of silibinin alone, increase the ROS production, and also increase the lipid peroxidation. Then a good results of the silibinin when the extract is encapsulated, the molecules is encapsulated inside the nanoparticles. In conclusion, from our results, in vitro and vivo results, nanoformulations are able to improve technological biopharmaceutical characteristic of extract of all singular molecules, can improve permeability, can improve the stability of the compounds, and reduce the toxicity of the molecules because the carrier protect the cells from the toxicity of the drug, and the formulation are useful for all route of administration, oral, eye application, or parenteral administration. It can be used also not only for pharmaceutical field, in the pharmaceutical field, but also in food, dietary supplements, or cosmetics field. Some drawback, some limits of the study reported in literature is the scale up in the production of these carriers, and also the evaluation of long-term stability and the cost efficacy in the production of these formulations. Thank you for your attention. If you have a question,
Thank you, Dr. Bergonzi, for your lecture, for showing us your excellent results from all these works that you have performed during these years. Now we have time for some questions, if the students have any. You can chat on here and we can translate them. I have a question, but it's only a curiosity. This last work that you showed us about silibinin and oleanoic acid, these nanoparticles that you made in your laboratory, have you considered to evaluate the hepatoprotective activity for this association? Because both compounds are hepatoprotective. Especially this flavonoid from Silybum marianum.
Yes, now we are performing tests with hepatocellular HEP2 to test the, for example, inhibition of lipid accumulation characteristic of. We are performing now the study with my colleague.
Okay. Because
I haven't the results at the moment.
The first thing that comes in my mind when I hear this this compound. There is a question here. Which are the most difficulty to incorporate extract in microemulsions?
Sorry, which?
Which is the most difficulty to incorporate extracts in microemulsions?
The difficulty to formulate in general an extract is due to the presence inside the extract of different compounds with different chemical structure, but also with different solubility, permeability. The extract is a mixture, no one molecules. And then it's difficult, and for two reasons, microemulsions with oil, water, and high amount of tenside is one of the best formulations to solubilize different compounds.
I can imagine natural products have a wide range of polarities, and some of them have a low, others have a higher solubility, yes, and so on.
Yes.
About this extract of olive leaves that you made as well. I don't know if I missed a part. This extract has different concentrations of these triterpenes. these
These are
compounds?
enriched. Is an extract produced by a Spanish industry with high amount of triterpenes. In the European project are involved two universities, Florence and from Germany, but many industry. And the principal investigator was an industry that produced a different type of extract from olive leaves. And
Okay.
I receive olenoic acid pure because is a percentage, the purity percentage is 98, and also enriched extract of triterpenes, about 70% of the triterpenes.
And these leaves are a waste. Oh, these leaves are a waste for the industry of olive oil because there are so many.
Yes. There are many industries that for because in Spanish, also in Italy, olive oil is important part of economy.
I can imagine. Alguma outra pergunta, pessoal? Well, that's we thank you again for your availability to perform a lecture for us, Dr. Bergonzi, and thanks in advance and good work.
Thank you very much for this opportunity. Bye.
Bye.
See you.
Thanks.
See you. Bye.